My name is McGowanM, and this lecture capture video is about two-dimensional nuclear magnetic resonance methods and how those methods can be used to examine a DNA structure. We're going to start with a little overview here. First, I'm going to introduce DNA structure, from the nucleotide bases to the sugar phosphate backbone. Then I'm going to go into NMR methodology. We're going to talk about how to process data using NMR software. And finally, we're going to figure out how to analyze data using a nosy walk. The first step is talking about deoxyribonucleic acid, or DNA. DNA is a double helix. There are two strands of DNA that wind around each other. DNA can essentially be broken down into three main parts. First, the nucleotide bases. Shown in green in the figure, nucleotide bases hydrogen bond to each other, which connects one strand of DNA to the opposite strand. The hydrogen bonds are the dashed black lines in the figure. Second, we have ribose and deoxyribose rings. These five-membered rings are shown in blue and red and can be differentiated by their name and structure. A ribose ring has two hydroxyl groups. A deoxyribose ring has only one, hence deoxy. RNA has ribose rings, and DNA has deoxyribose rings. These ribose or deoxyribose rings are the first part of the DNA backbone and connect the nucleotide bases to the phosphate groups. This phosphate group is the third part of the DNA structure. The phosphate groups in the figure are the solid red and blue circles. Each one is a phosphorus atom with 4 oxygen atoms bonded to it. These phosphate groups are the link between ribose and deoxyribose rings and form the second half of the sugar phosphate DNA backbone. This sugar phosphate chain links each nucleotide together, regardless of the identity of that nucleotide. So, no matter the sequence of DNA, you can bet that there will be 2 sugar phosphate chains that hold the double-stranded DNA in place. Now let's move on to the nucleotide bases themselves. As discussed, the first part of the DNA structure is the nucleotide bases. Each strand of DNA has a sequence of nucleotides that hydrogen bond or base pair with the nucleotides on the opposite strand. Each base pair will be a purine, guanine or adenine, paired with a pyrimidine, cytosine or thymine. Specifically, guanine pairs with cytosine and adenine pairs with thymine. In RNA, there is actually a fifth base called uracil, which replaces thymine. Therefore, in RNA, adenine pairs with uracil and not thymine. However, this research focuses on DNA, so that is what this video will focus on as well. As previously mentioned, the base pairs are held together by hydrogen bonds. So to summarize, an example sequence of DNA, ATCG, is just the 4-letter initials for a sequence of adenine, thymine, cytosine, and guanine. Now let's get into the sugar phosphate backbone. As we discussed before, the sugar phosphate backbone connects the nucleotide bases together and is independent of the nucleotide sequence. Now we are going to zoom in on this backbone to see the specific structures. The sugars bind to each nucleotide base via the open nitrogen atom. You can see the example here of both a purine, adenine, and a pyrimidine, cytosine, and which nitrogen they bond to. This sugar has a very specific numbering scheme that starts with the carbon atom bonded to the nitrogen of the nucleotide base. We number the ring with primes because the bases have the priority numbering scheme. So, the numbering goes like this. The carbon bonded to the nitrogen is labeled as the 1' carbon, and then we go 2', 3', and 4' on the ring, then up to 5' on the CH2 group that is bonded to the ring. This numbering is very important because we can identify hydrogens that are connected to these carbons by their numbering. The one exception is the 2' carbon, which has 2 hydrogens bonded to it. The second hydrogen, closer to the next sugar, is deemed the 2' hydrogen. The last part of the sugar phosphate backbone is the phosphate group. The phosphate group is bonded to the sugar through the 5' carbon and the 3' carbon. This numbering scheme lends itself to the directionality of DNA. This so-called 5' to 3' direction indicates that the addition of successive nucleotides starts at the 5' carbon, and then the next phosphate group is connected to the 3' carbon. DNA always goes in the 5' to 3' direction. This is what the overall structure of DNA looks like. The strand on the left goes from 5' to 3', as we discussed, and base pairs to the second strand through complementary nucleotides. The second strand goes from 5' to 3' as well, which is why it appears to be upside down. Now that you have an overall sense for what a DNA sequence looks like, we can get started talking about how this plays into your research. The Dickerson dodecamer is the sequence of DNA that you are going to start your research studying. We use this sequence because it is well known and the literature values are known. The sequence itself, CGCGAATTCGCG, numbered 1 to 12, looks like this. What we are specifically going to look at is how the interactions between each nucleotide base, its 1', 2', and 2" hydrogens, and its neighboring bases' hydrogen atoms go together on a nuclear magnetic resonance spectrum. When hydrogen atoms are near each other in physical space, they interact with each other and resonate off of each other. When hydrogen atoms are further from each other, like in the image on the right, that interaction is absent, and we can actually measure those interactions through nuclear magnetic resonance. Nuclear magnetic resonance, or NMR, is an analytical method that focuses on the magnetic properties of atomic nuclei, in this case hydrogen atoms. When atoms are close to each other in space, the intermolecular magnetic field changes the resonance frequency, and this change can be graphed. One-dimensional NMR focuses on the differing chemical shifts to distinguish between hydrogens in an organic molecule. These different chemical shifts are caused by a chemical environment, aka what other atoms are close by, those atoms' electronegativity values, and shielding. One-dimensional NMR is useful for determining the structure of an organic molecule, but for an incredibly complex compound like DNA, we need a more powerful analytical tool. This is where two-dimensional NMR comes into play. As opposed to chemical shift, 2D NMR is based on dipolar coupling. The two dimensions are two frequency axes that represent a one-dimensional chemical shift. Essentially, a single 2D spectrum is generated by a series of 1D experiments, and the resulting plot shows cross peaks that connect resonances from hydrogen atoms that are spatially close together. For example, a nucleotide base hydrogen to its 1', 2', or 2" hydrogen. We can use this 2D NMR to solve the puzzle, so to speak, and identify a sequence of DNA based on the location of these cross peaks. We will get more into how that process works a little later in the video, but for now it is enough to understand why the cross peaks are located where they are. The location of these cross peaks brings us to the chemical shift ranges that will be helpful to us in this research assignment. On this chart, we will be looking at the blue lines in the middle for DNA. These short lines indicate, in parts per million, where we can find those hydrogens on an NMR axis. We will be focusing mostly on the H8 and H6 hydrogens on the nucleotide bases, and the 1', 2', and 2" hydrogens on the nucleotide's sugar group. Now that we have a basic understanding of DNA and NMR analysis, we can get started analyzing the Dickerson dodecamer. Now we're going to start analyzing data. The first step is to open ACD NMR Processor, the software we will be using for the duration of this process. Then we're going to go to File, then Open to find our data. Now, since I've already been using this data set, it's already here for me to open, so I'm just going to hit OK. Once you have the data set open, make sure that the plot style is set to contour, not intensity, and you can see this up here at the top left of the screen. You'll notice that the data doesn't appear to have peaks. There are just a lot of randomly placed dots. Before we can analyze any peaks, we must process and fit the data. Step 1 of this process is a full Fourier transform. So we're going to click Full FT, the button at the top left of the screen, and then we're just going to hit OK, and the software is going to do it for us. The next step, we're going to go up to Process, then down to Reverse Spectrum, and then we're going to click along F1. Basically, this just reverses the spectrum along the axes. Step 3, we're going back to process, this time down to phase correction, and we're just going to hit auto, and again the software is going to do the phase correction for us. Now the last step of data processing is setting a reference point at the water peak. For most experiments, the water peak is set by default to 4.62 ppm. However, the water peak is actually temperature dependent, and in this experiment, the water peak needs to be set around 4.77. So in order to find this water peak, we're going to zoom in on the center of the spectrum here. We're going to go to around 4.62, which you'll see at the bottom of the screen, and there we have our reference point for water. So now that we found the point, we're going to go up to Analysis, down to Reference, then we're going to go to Enter Mode. In Enter Mode, all we have to do is click on the point that we want to set as the reference, and then it'll come up with this nice dialog box. And then under New Point, we're going to type in 4.77 for the new point of F1 and F2. Then we're just going to hit OK. Now our data is properly referenced, and we are ready to analyze our data. So now we just hit the green check mark to save all of our changes, zoom back out, and now we're ready. Now that we have our data processed, it's time to start analyzing it. Our main tool in analyzing a sequence of DNA is going to be a sequential walk. A walk is a step-by-step analysis that leads to the correct labeling of peaks by knowing what protons are close in space via dipolar coupling. There are a couple of different walks depending on which region of the NMR spectrum we are discussing, but for the purpose of this research, we will be mostly looking at the aromatic to 1' sugar region and the 2' to 2" sugar region, as these are the regions that can be used to identify a particular sequence of nucleotides. Since we already know the sequence, this video mostly serves to acquaint yourself with the process of peak identification and the nosy walk. The first step in any identification is unique features, so that is where we will begin as well. We know from our nucleotide structures that thymine is the only base to have a methyl group, so that is one important identification tool, as the only thymine bases in the Dickerson dodecamer are the T7H6 and T8H6 bases. Methyl peaks have a chemical shift around 1 ppm, and we are looking for where those peaks indicate an interaction with the nucleotide bases, which is around 7 ppm in the aromatic region. On the spectrum, this area is in the top left and is known as the 2' to 2" region. So let's zoom in on that. In this region, and in the 1' region, the bases usually have the following shifts. Thymines on the right side, most upfield, C's and G's in the middle, between 7.1 and 7.8 ppm, and A's on the left side with the most downfield shift. Remember, we are looking for peaks in a cross area where we would expect to find the T7H6 and T8H6 base protons interacting with the T7 and T8 methyl group. So we look on the vertical axis around 1 ppm, cross that with the horizontal axis around 7 ppm. That gives us these two peaks right here. Now the question is, how do we distinguish between T7H6 and T8H6? Well, we know that the Dickerson sequence goes A6, T7, T8. So we can perform a methyl walk to determine the location of bases A6, T7, and T8. The T7 methyl group will interact with the A6H8 base proton, which we predict is located somewhere close to 8 ppm. This tells us that of the two methyl peaks we have found, the top one has to be the interaction of the T7H6 base proton on the x-axis and the T7 methyl on the y-axis, because if we draw a horizontal line across the spectrum, there is another peak in the A range, specifically the A6H8 base proton interacting with the T7 methyl. So now we have found the A6H8 base proton and the T7H6 base proton, just because of our identification of the T7 methyl line. Going back to these two methyl peaks that we found, we propose that the bottom one now has to be the T8 methyl on the y-axis because we already found the T7. This peak right here is telling us that the T8 methyl on the y-axis is interacting with the T7H6 base on the x-axis. So if we draw a horizontal line across the T8 methyl, we run into another peak, which deductively has to be the T8H6 base proton interacting with the T8 methyl. Now we have found bases A6H8, T7H6, and T8H6. Let's summarize what we learned from this short methyl walk. 1. The x-axis shows the nucleotide base protons, so if a vertical line was drawn through multiple peaks, then those peaks must all be interactions of different hydrogen atoms interacting with that nucleotide base. 2. The same thing goes for the y-axis. If a horizontal line is drawn across the spectrum, then any peaks are the interactions of a methyl group or a single hydrogen atom interacting with multiple bases. 3. We can use the sequence of the Dickerson dodecamer to walk from base to sugar to base and identify all the peaks, and ultimately verify the sequence. Now these peak assignments are all based on trends, and they still need to be compared to and verified by literature values. The next step after the methyl walk is the 1' hydrogen walk. In this walk, we can use the knowledge of the A6H8, T7H6, and T8H6 locations in the 1' hydrogen region to identify the rest of the base locations and their 1' hydrogen locations. The 1' region is located directly under the 2' to 2" region, so the same x-axis but a shifted y-axis, from about 5.25 to 6.25 ppm. We can start at T7H6 or T8H6 because we already know the shift of those bases. From there, we can walk from the T7 base / T7 1' peak to the T8 base / T7 1' peak by following the T7 1' horizontal line. Then we can follow the T8 base vertical line to the T8 base and T8 1' peak, and so on and so forth until we complete the walk. Now there are a few tricks that can help when making peak assignments. 1. Cytosine's inner base peaks are generally the most intense peaks, so that can help you distinguish them from the thymines. 2. The A5H8 and A6H8 bases are potentially overlapped, so there will only be one vertical line for both of them. 3. When you finish with the 1' region and move on to the 2 to 2" region, it is important to note that a walk can only be done for the 2" peaks and not the 2' peaks. However, you can distinguish between them because generally the 2' peaks are more upfield than the 2" peaks. And here's just a trick for you. It can be really helpful to print out the spectra and use a ruler to draw lines between bases and to scribble notes on, while also having the spectra on the computer screen to get a closer look if needed. This almost dual screen option is really helpful when trying to distinguish between two very similar bases. Lastly, it's a great idea to keep a running table of peak assignments just so that you don't get confused when you're making your peak assignments. And that is all I have for you, so good luck, puzzlers, and may the chemistry gods look down upon you to help you with your task. Happy researching!